Good morning, everybody. I'm just here to introduce Shannon and Priscilla, but I thought, especially for the younger ones here, I'd just briefly introduce our field, because our actual field of research and where we were all trained is in clathrin-mediated endocytosis. And it's a little bit of a different field. So clathrin-coated vesicles were originally discovered by Barbara Pierce, who worked at the LMB in Cambridge. And that work was expanded by Marlon Farquhar and Catherine Howell. And so what you can see straight away is that the three women who started our field are all senior women in science. So Barbara Pierce discovered clathrin-coated vesicles, and her PhD student and then postdoc was Margaret Scott Robinson, who discovered adapter proteins. And Marlon Farquhar and Catherine Howell both trained a young lady for her PhD and postdoctoral work called Jenny Stowe. And in turn, I was trained by Margaret Scott Robinson, and Shannon did her PhD with Professor Jenny Stowe. And so firstly, the field is very much dominated and has been over time by women: Sandra Schmidt, Gillian Griffiths, Frances Brodsky. And it's always been an exceptionally friendly field, and it's set up as family. And once you're a member of the family, you're always a member of the family. And I think this was really demonstrated recently when both Laurette and Priscilla presented talks about our work in Cambridge. And lots of people sent photographs back of the grandchildren's visit to the department. And they both came back saying they were literally treated as grandchildren and parts of the family. So I thought it was just a nice background to the field that we work in. And Shannon and I will have worked together for 10 years at the end of this year, and we are the grandchildren of Barbara Pierce. So Shannon did her PhD with Professor Jenny Stowe over at the IMB. She then moved north and was in charge of hospital safety for many years in the middle of Queensland, and then returned to the lab where she has remained for nearly 10 years. And she also, at the same time, has 3 absolutely beautiful children, and is amazing at cell biology, imaging, anything microscope related. The next presenter after Shannon this morning will be Priscilla Oliveira de Lima. So Priscilla's a final year PhD student in the lab. She took 4 days to reach the lab from Brazil. Her first task on hitting the lab was to be the only student allowed to be involved with our business contract with Merck. And so Priscilla's introduction was to literally nearly kill herself hitting the deadlines for the company, getting the report ready. And she did an amazing job of that. Since then, she's done things like won the EMBL scholarship, traveled to the EMBL in Germany, where she also won the poster prize, and has given talks around the world, and she's amazing as well. And so given these 2 ladies are so exceptional, I'm not going to say anything else. I'm just going to go to the first slide, introduce the problem, and let Shannon and Pri take over. You can share screen, Shannon. You're on mute. Okay, so the lab works on reversing cancer resistance by manipulating cellular trafficking. The problem that we had is that 30% roughly of patients respond to targeted therapies such as cetuximab or trastuzumab. And that's also becoming clearer for checkpoint therapies such as anti-PD-L1 and anti-PD-1. Next slide, Shannon. So this is the problem we approached, that targeted therapy, antibodies only work in 30% of the patients. And if you look at the next slide, Shannon, just for something like cetuximab alone, the therapies cost $30,000 to $60,000 per patient. In the case of the checkpoints, it was 110. And the cost of non-responders to the Australian system just for cetuximab is $355 million per annum. And so in combination, that can probably be approved. And the cost of non-responders or recurrence and chronic side effects of the checkpoint inhibitors to Australia has not yet been calculated, but imagine it's going to be enormous. So this is the problem that we started addressing, and I will hand over to Dr. Joseph.
Shannon, just before you start, have you got two screens going?
Yes, I do.
You're just going to swap your slides over to the other one just because we're seeing the presenter view.
Yeah. Are you seeing the presenter view?
Yeah.
Oh, okay.
So just put your slides on the other screen and it should work okay.
Is that all right now? Or
nope, still the same.
Okay. I'll just stop sharing for a second.
Yeah.
And come back to you. Let's just turn that off. Okay. Okay, sorry everybody. Okay, now that should work now. Okay, is it working?
No, still on presenter mode.
Okay, let me just, let's go. If I move this one up here. Is that better? Can you see it then?
Nope, still presenter mode.
Oh, okay. Okay, let me just.
Can you turn off the presenter mode?
This just worked before when we did it. So
I know it did, yeah.
Shannon?
Yep.
See up next to the X that you were about to press?
Yep.
Try the one next to it.
The restore down.
Oh no.
End slideshow.
Display settings. Does anybody know?
I don't. Okay.
Have you tried putting your slides on the other screen that you've got?
That's what I just did. I've got Zoom and everything on the big screen.
Yeah.
There's a few options at the top that has fit the window. Maybe that's just for mine.
No, on presenter, on slide show, there's a tick box for use presenter view.
On slideshow, there's a tick box for use presenter.
Should be able to untick it.
So if you come out of...
Swap presenter view and slideshow.
There we go.
Is that right now?
Not.
No.
There we go. Yep.
Yay. Okay. Sorry, everybody. Technical issues. Okay. Let's get started. So as Fiona said, this project started as a question from the clinicians wondering why, even though over 90% of the patients expressed the target of the drug, only less than 30% of the patients were responding. So in order to tackle this problem, what we thought we'd do was look at the actual drug target, and that's the EGF receptor. So in order to do this, we took patient biopsies back to the lab. We incubated them with fluorescent EGF ligand. So the EGF ligand binds to the EGF receptor, and it actually binds at the same site as cetuximab, the drug. So it's as a surrogate marker for the drug binding site. So what we found when we did our EGF ligand uptakes, we fixed the cells and analysed them by microscopy, and this here is a super resolution image. And what we found was that there were two patterns of drug target. We had EGF ligand that was blocked on the plasma membrane, And then we had EGF ligand that was internalized into the cell. And then when we did the blind correlations of the patient outcomes, we found that those patients that had the blocked EGF ligand were the responders, and they were the ones that had tumor aggression. Those that had the internalized ligand were the non-responders with some stable disease. So this suggested to us that the EGF trafficking status correlated to the patient outcomes for the treatment with the anti-EGFR monoclonal antibody therapies. So this led to the major lab question: can we turn a tumor non-response pattern into a tumor response pattern? And how can we do this? So yes, we thought we could, if we could inhibit a molecule called dynamin in patients. So the EGF receptor comes in classically and predominantly via the clathrin-mediated endocytic pathway. So you have ligand binding, the receptor gets clustered, it signals, the clathrin machinery is recruited, you get invagination from the membrane, and the final step is the scission of this vesicle, this invagination from the membrane into a vesicle. And this is catalyzed by the protein dynamin. So we figured that if we could stop the process of this scission, we would increase the target on the cell surface, and this may lead to the change to that responder pattern and therefore better outcomes for patients. So this was the premise for the paper that we've recently had published in Cell, Endocytosis Inhibition in Humans to Improve Responses to ADCC-Mediating Antibodies. We have a number of first authors. It occurred over many years. We have significant collaboration within UQDI and with the PA hospital and also with external partners. This is a current picture of the lab. Priscilla and I have the pleasure today of presenting on behalf of the author list. So thank you for that opportunity. So I'm going to be talking about the in vitro data, I suppose, that supported the progression to our animal models and also to the human trials. And that's what Priscilla is going to be talking about. So this is a lovely Priscilla here. So first up, just a little bit about monoclonal antibodies. So cetuximab is an anti-EGFR monoclonal antibody. It's humanized, meaning it has a humanized IgG1 domain here and a mouse Fab domain which binds to the EGF receptor. It's thought to work by two main mechanisms. It's Fab-dependent mechanisms of action, which involve downmodulation of target antigen, inhibition of signaling, and together these are thought to reduce tumor cell proliferation, induce apoptosis, and reduce angiogenesis. And the second major role is our Fc-dependent mechanisms of action. So this is what I'll be talking to you mostly about today. So this is the process where the antibody induces Fc gamma mediated cytotoxic immune responses. So this is the binding of the antibody to our epitope. This then recruits the effector cells or natural killer cells, which bind via the Fc gamma R receptor. It's a threshold receptor, meaning it needs a level of signal before it can kill the cells. So it secretes granzymes, perforins to kill the tumor cell. So we can measure this using our antibody-dependent cell-mediated cytotoxicity assay in vitro. So we started the project looking in vitro to see if we could actually manipulate the EGFR trafficking within the cell. And in order to do this, we used a drug, dingo-4A, which is an inhibitor of dynamin. We used initially two different cell lines. We've got cetuximab sensitive cells, the A431 cells, and cetuximab resistant cells, KJD cells. So when these cells are treated alone with cetuximab, it does not affect the KGD cells, but it inhibits cellular proliferation in the A431 cells. When we incubated them in the presence of dynamin, the dynamin inhibitor dingo-4A, we could see an increase in the cetuximab on the plasma membrane. This persisted with the different time courses. This was also evident for our resistant cell lines. We saw an increase in cetuximab on the cell surface. We then looked at the levels of antibody-dependent cellular cytotoxicity, or ADCC, and to do this we used an optimized assay that we did with the Wells labs. And basically what we do here is we incubate labeled tumor cells with immune cells and our combination of drugs, and then we can assess for specific tumor cell death. So what we found in the sensitive cells was that we had nearly 40% cellular killing in the presence of immune cells and cetuximab, and this could be further increased with our dynamin inhibition. And this was the same in resistant cells. We saw a significant increase in cell death. So together, these experiments showed us that we could increase the surface levels of our drug by using our dynamin inhibition, and this in turn led to increases in ADCC. So we would expect that if we were increasing the surface levels on the cell surface, that this would be applicable to all endocytic mechanisms. So we thought we'd test this by using clathrin inhibition. So we used pit stop as a clathrin inhibitor. And what we found was that pit stop, although it inhibits internalization of the target, it did not cause this increase in ADCC. And this was evident in both our cetuximab sensitive and our cetuximab resistant cells. And the reason for this is that it's not just the total surface levels of EGFR, but rather the clustering of the EGFR cetuximab complexes that promotes the ADCC. And this was evident in one of the papers we published together with the Robinson and McCluskey lab, where we showed that we could see an increase in the surface levels of the EGF receptor, but this didn't correlate to an increase in the EGF ligand, in that the EGF ligand wasn't clustered enough for us to see in the presence of the clathrin inhibitor, but it was with the dynamin inhibitors. So we then wanted to ask if this therapy was applicable to more than just cetuximab. So we looked at additional monoclonal antibodies. So the first one we looked at was trastuzumab. So trastuzumab is an anti-HER2 monoclonal antibody, and it's used typically in breast cancer against HER2-positive breast cancers. It's also, HER2 comes in via the clathrin-mediated and FEME pathway, just like EGFR. It's expressed in both our SCC cell lines, and our breast cancer cell lines are here on the bottom. And what we found was that we could indeed increase the ADCC level of trastuzumab-mediated ADCC with dynamin inhibition. You can see that here in the breast cancer cell lines, and this also occurred in the SCC cell lines. What's significant about this is that the cells that expressed more HER2 had were more responsive to the increase in ADCC with trastuzumab than those that expressed EGFR, which were more responsive to cetuximab-mediated ADCC. So this showed us that the combination therapy was specific to the receptor expression. It wasn't just a general process mediated by the immune cells, but was specific for the tumor cells. We also looked at lumratuzumab and daratumumab, which are monoclonal antibody therapies that are still undergoing trial against ERBB3 and IGF-1R respectively. And what we found with these two monoclonal antibodies was when we treated the cells, they were rapidly internalized. When we treated them with both PCZ and Dingo4A, we did not block this internalization, which suggested that these two drugs came in via dynamin-independent pathways. And when we did our ADCC assays, we found very little ADCC, and this could not be increased with PCZ treatment. So together, this result showed us that firstly, we needed the proteins to be stuck on the cell surface, or that block in the internalization to see ADCC. And secondly, it showed us as an IgG1 isotype control that it's not just due to the clustering of the IgG on the cell surface, but it's specific for both the protein that it's binding to and also the endocytosis inhibition. So we also had a question about whether it was due to inhibition of dynamin in the tumor cells, or was it due to inhibition of dynamin in the immune cells? So in order to address this, we transfected our tumor cells with dynamin K44A dominant negative mutant. So this point mutation allows dynamin to self-assemble at the cell surface, but it blocks its internalization. So we transfected the cells, we FACS sorted them, and then we put them in our ADCC assay with cetuximab, or with cetuximab in the presence of PBMCs. And you can see on the right here that our K44A mutant in the presence of immune cells and cetuximab caused significant ADCC, where our mock transfected cells did not. So this confirmed that the ADCC is dependent on dynamin inhibition in the tumor cells and not dynamin inhibition in the immune cells, because these cells were not transfected with that mutant. So in our process of translating this to the clinic, we knew that we couldn't use our Dingo 4A. It's not able to be used in patients. So in collaboration with the McCluskey lab and the Robinson lab, we published a paper that found that there were potent dynamic inhibitors already used in the clinic. And these are the phenothiazines. So they inhibit the dopamine receptor, but they're also endocytosis inhibitors as an off-target effect. They've been in clinical use for 30 years at quite high doses. And in collaboration with the clinicians, it was deemed the most safe and potent among the antipsychotics was prochlorperazine, better known as Stemetil, and I'll probably refer to it as PCZ as I talk about it. So the idea was that we could repurpose these drugs to be used in combination with our monoclonal antibody therapies to increase their efficacy. So we first had to check that the PCZ worked in our in vitro assays as our other dynamic inhibitors did. So we treated our cells with PCZ in the presence of EGF ligand, and we effectively caused that block in endocytosis. So if you look on the right panel here, you can see that the EGF ligand is in an endosomal pattern. When we treat it with the PCZ, the EGF ligand was blocked on the cell surface. And that was reminiscent of what we saw in those patient tumours when we first got them. We also found that PCZ increased cetuximab-mediated ADC in both our A431 cells, our sensitive cell line, and our KJD cells, our resistant cell line. We also wanted to know what this was doing to the EGF ligand during its endocytosis. So in order to do this, we set up a collaboration with Professor Rob Parton and James Ray over at the IMB, and we did electron microscopy of our treated cells. So as I said, EGF receptor typically comes through this clathrin-mediated endocytic pathway, but also comes through a number of other pathways depending on concentration of ligand and timing. So if you look in the left-hand side here, we have the control cells, and you can see that the EGF ligand is in these multivesicular, or these endosomes. And this is characteristic of EGF ligand internalization. When we treated the cells with PCZ and did electron microscopy, it resulted in a number of changes. Firstly, we had an increase in EGF HRP stuck in clathrin-coated pits at the cell surface. They're indicated by these red arrows here. We had reduced transport of EGF to the multivesicular endosomes, demonstrating that clathrin-mediated endocytosis is slowed down. And we had the appearance of these tubules emanating off the plasma membrane. And these were indicative of a block in non-clathrin-dependent fast endothelium-mediated endocytosis. So effectively what our PCZ was doing was slowing down the internalization of the EGF ligand. So we then wanted to look at how this translated to the interaction between our immune cells and our tumour cells. And so again, we've done electron microscopy here with the Parton lab. And what we've done here is we've incubated our tumour cells with our immune cells with and without PCZ in the presence of cetuximab HRP. So there's some striking differences here that I'd like to point out. So if you just look at the top panel here, I'll point out some of the differences. So on the left here, we have an immune cell. And so it's identifiable by these electron-dense cytotoxic granules, and you can see them in a number of the cells here. What you want to look at here is the immune synapse, and you can see that the cells are touching via these finger-like projections, or they've got a small region that's touching. And these were not common to find these cells within the population of cells that were analysed by the electron microscopy. And you can see here we have our cetuximab HRP in an endosome. Now, when we treated the cells with PCZ, we saw a striking change. You can see that the immune synapse, or that interaction between the tumour cell here and the immune cell here, has changed. Back we go. It's effectively zippered up along the membrane, and that contact is much more tighter. And you can see that in all these images here. It's actually really, really cool. And you can see here, we have this electron dense region here is actually our cetuximab HRP that is on the cell surface of the tumour cells. And you can see that that's at the interface, or the immune synapse, between the immune cell here and the tumour cell. So effectively what we've done here is we've increased the contact between the tumour cell and the immune cell. And we think that this is in a way playing a role in that increase in ADCC that we're seeing. The other question that we were hit with a lot is what happens to the signalling when we use the PCZ, when we bring the protein to the cell surface? So potentially the PCZ could inhibit the role of cetuximab. So we did directed an extensive Western blot analysis of the different signaling pathways in numerous cell lines under different drug treatments. And what we found was that the drug combination therapy didn't adversely affect cetuximab's ability to inhibit signaling in the cetuximab sensitive cells. So cetuximab inhibits phospho-ERK and phospho-AKT, part of the MAP kinase pathway and the AKT pathway. And we could see in the presence of cetuximab and PCZ that it retained its ability to inhibit these signaling pathways. What we found in the resistance cell lines was that PCZ actually inhibited AKT. So it repressed the signaling activation in cetuximab resistant cells. So in conclusion, our PCZ treatment didn't adversely affect the role of cetuximab in inhibiting signaling. So in summary from this in vitro data, this was the hypothesis that we had when we were going to our mouse models and our human models. So in scenario 1 here, we have the target of our monoclonal antibodies inside the cell. Therefore, the drug cannot bind and we get no engagement with the immune cells and no immune response. In scenario 1, we have some of the drug target on the cell surface. We have binding of the monoclonal antibodies, recruitment of the immune system, but there's not enough signaling through this receptor to induce an immune response. In scenario 3, we have addition of our dynamic inhibitor. It causes the receptor to cluster. We have recruitment of our monoclonal antibody, binding of the FC receptor, signaling, release of perforin granzyme to kill that tumour cell. And so now I'll pass you over to Priscilla, and she's going to go through the mouse models and the human trials. So I'll just stop sharing my screen, and then Priscilla, you right to go? Thank you, and I'll be back at the end.
Thanks so much, Shannon.
Okay. Yeah. Can you see it? Yes. Okay.
Hi everyone, I'm Priscilla. Sorry. Okay, I can just put my headphones. That's all right. Hi everyone, good morning. I'm Priscilla, and as Shannon has already introduced me and Fiona as well, I'm a third-year PhD student in Simpson Lab, and today I'm going to talk to you about our in vivo data. So first, we wanted to look at the effect of PCZ or prochlorperazine in the relocalization of receptors in KJD xenograft tumors. Especially because, as Shannon mentioned, KJD cells are resistant to cetuximab therapy. So we did an IGF-48 uptake, and as she also mentioned, we use EGF48 as a surrogate marker for cetuximab binding sites. And we do this uptake prior and post injection with prochlorperazine. And we can see here, yes. So we can see here that prochlorperazine relocalized the drug target to the cell surface in these xenograft tumors. So then we moved to the in vivo studies, and we challenged NSG mice with A431 cells that are highly EGFR expressing cells and sensitive to cetuximab. So we injected these mice with human NK cells, and we treated them with Fab fragments, cetuximab Fab fragments, which basically, as Shannon explained, binds to the receptor and inhibits downstream signaling pathways that are involved in cell survival and proliferation. And we also treated another group with full-length cetuximab. So we not only have inhibition downstream signaling pathways, but we also have induction of antibody-dependent cell cytotoxicity through interaction of the FC, part of the FC from the receptor. We have interaction of the FC portion of the antibody with the FC gamma receptor and NK cells. So what we see with this experiment is that even though we can achieve stabilization of tumor growth by inhibiting these signaling pathways, without induction of ADCC, we cannot achieve tumor clearance, and these tumors actually, tumor growth rebounds. So then we move to another in vivo experiment where we challenged NSG and NSGA2 mice with KGD cells. And as Shannon mentioned, KGD cells are resistant to cetuximab therapy. And we treated these animals with either cetuximab or cetuximab plus prochlorperazine. So we see that in the presence of PBMCs, we see that the animals treated with cetuximab only, they show stabilization of tumor growth, while animals treated with combination therapy, cetuximab plus PCZ, they show tumor clearance. And not only that, but these animals did not show any lung metastases, while all the untreated animals showed lung metastases, as you can see here on the figure. So then we asked, can we test, is it going? No. Sorry. So then we asked, can we improve the efficacy of a different monoclonal antibody for a different target? So in a contract with Merck KgaA, we tested the efficacy of their antibody avelumab, which is an anti-PD-L1 antibody, in combination with PCZ in syngeneic murine model of colon carcinoma using CT26 wild type cells. And we can see that combination of avelumab plus PCZ increased ADCC in vitro. So then we tested also in CT26 xenograft tumors, and we can see that PCZ, the treatment with PCZ, also relocalized PD-L1 to the cell surface in these xenografts. So we challenged BALB/C mice with CT26 wild type cells, and here you can see the different treatment arms. We have the control or untreated mice, and then we have the group that was also treated with, prochlorperazine only. And we see that PCZ alone has no significant effect when compared to the control group. So it was also used as a control. We see here in the groups treated with Fab fragment of avelumab and Fab fragment plus prochlorperazine, we observe stabilization of tumor growth, but at a certain point, this tumor rebounds, so they keep growing. So this indicates that the inhibition of the PD-1, PD-L1 axis is not enough to promote, in the majority of these tumors, it's not enough to promote a full tumor clearance. So we have these tumors progressing. When we treated these animals with full-length avelumab, we see that the tumors kept progressing despite the treatment. However, when we treat these animals with our combination therapy, avelumab plus prochlorperazine, we see that 50% of these animals showed full tumor clearance. And this is statistically significant when compared to the controls. And so this was very interesting because these results show that PCZ, when we add PCZ, we can increase the drug target, in this case, PD-L1 availability on the cell surface for avelumab binding. And this way, we're not only inhibiting the PD-1, PD-L1 axis, therefore resulting in T cell activation, but we are also enhancing ADCC by stimulating or promoting stronger interaction with these NK cells. So we can enhance avelumab-mediated ADCC. Another thing that was interesting about this experiment was that the treatment did not affect the mice body weight, but we did have one side effect in the groups treated with Fab fragments. We see that these mice treated with Fab fragments, they actually started showing lethargy, and they actually died after the fourth injection with Fab fragments. And the necropsy we performed showed that symptoms that were similar to an autoimmune disorder, so suggesting that these animals raised antibodies against the Fab fragments, resulting in their death. But another thing that was very interesting about this experiment was that we took these animals that showed tumor clearance and we re-challenged them with CT26 wild type cells. And we see that these mice that previously showed tumor clearance, they did not develop any tumors for over 300 days in comparison with the naive mice, suggesting that this increased avelumab-mediated ADCC can induce an immune cell memory in these models. So the next step is to investigate the mechanism by which this immune cell memory is being developed in these models. So to observe, we wanted to observe the exact time point when these targets are moved to the cell surface. So we conducted this experiment and we challenged biopsy mice with CT26 wild type cells, and when tumors reached 0.3 to 0.8 centimeter cube, we administered PCZ at 0.8 mg per kilo IV. And then at different time points, we collected tissue and we sent this tissue to pharmacokinetic analysis. And half of the tumor we actually performed PD-L1 labeling. And here you can see that we see an increase of the PD-L1 labeling from 5 to 10 minutes, but we see accumulation and this peak in the 30 minutes and 1 hour time point post injection with prochlorperazine, as you can see here. We see that 8 hours, as the PK analysis showed that the half-life of PCZ in the tumors was 6 hours, we can see that in 8 hours, these surface levels are resetted, so they return to normal. A point that I would like to make about this experiment, or in general, is that we know that endocytosis inhibition is a temporary event in this case, and these, after the effect of PCZ wears off, when the effect of PCZ wears off, what we have is this large and rapid wave of endocytosis. And this strategy could actually be used to improve the efficacy of antibody drug conjugates. As you have, we can promote by this strategy an accumulation of these antibody drug conjugates on the cell surface. So when these the effect of PCZ wears off, you have like a huge endocytosis, wave of endocytosis of these highly payloads of antibody drug conjugates, which could improve their efficacy. That's very interesting. So then we move this to a proof of mechanism phase 1 trial in patients' tumors. So what we did was we collected tumor biopsies of patients prior and post-infusion with prochlorperazine, and we did an EGF uptake to observe localization of receptors. And we see that prochlorperazine relocalizes drug targets to the cell surface and reverse the heterogeneity of these receptors in these tumors. So we started a phase 1b safety trial in recurrent and metastatic head and neck squamous cell carcinoma and triple negative breast cancer patients with our combination therapy of cetuximab plus prochlorperazine. So these patients received cetuximab at a loading dose of 400 mg per meter square and a maintenance 250 mg per meter square, with administration of prochlorperazine at 0.8 mg per kilo, 48 hours post-loading cetuximab and 1 hour post-maintenance cetuximab. So then after 6 weeks of treatment, of combination therapy treatment, these patients were assessed by CT and PET-CT imaging, and if they showed stable disease or a better outcome, then they received this for further 6 weeks of treatment. If these patients showed progression of disease, then they had to withdraw the study. And we have recently completed this study, and we actually treated 12 patients with relapsed head and neck SCC or adenoid cystic carcinoma or EGFR overexpressing triple negative breast cancer. And we gave these patients the combination therapy for 57 weeks in total. And we see no adverse events or grade 3 or 4 toxicities and no hematologic toxicity. We only saw grade 1 sedation, akathisia, and hypertension. That was, these effects were related to PCZ, but they were resolved within 24 hours. We see that 50% of the patients developed grade 1 cetuximab related rash. And we know that the rash correlates with positive therapeutic response. So this is very exciting because we know that while cetuximab monotherapy has a response rate of only or about 15%, our combination therapy achieved a rash in 50% of the patients and 60 of those who actually completed a minimum of 4 weeks of combination therapy. Here you can see this 29-year-old male with metastatic head and neck with pulmonary metastases before undergoing the clinical trial. So you can see here the mets in the lungs. And after 6 weeks of treatment with our combination therapy, we see evidence of efficacy, and this is very exciting. So now what we're gonna do is, what we're doing is to, we want to expand this approach and this strategy and test the therapy, the combination therapy of prochlorperazine and with other antibodies for other diseases, so we can include other patients in our future trials. So for example, we have been establishing and developing preclinical models to test our therapies. For example, for perineural invasion of cutaneous SCC in the head and neck region, which is a form of metastasis where cutaneous SCC actually invade nerves. And we have also establishing, developing preclinical models of adenoid cystic carcinoma. I'm currently working in the perineural invasion project, and I'm just going to talk briefly. We have established at UQDI 2 different models to test our therapies for perineural invasion. Here, this is the first model. This is the sciatic nerve model, as you can see, and this has been established previously by the board et al. From the Sloan Memorial Kettering Center in New York, and I personally went there and learned this model from them. So this model is interesting because we can inject cancer cells directly into the sciatic nerve of animals, and then so we ensure that there is 100% of nerve invasion. So when we can treat, then we can treat these animals with our combination therapy. But this model does not reflect the disease, which is the nerve invasion of this cutaneous SCC into nerves in the head and neck region. So we have also established at UQDI another model in collaboration with Professor Glen Boyle from QIMR and our whisker pad model. So in this model, we inject cutaneous SCC into the whisker pad of mice, and we perform primary tumor resection when tumors grow. And then we keep these mice alive 2 weeks post-surgery, and we can analyze through live imaging progression of disease and euthanize these animals, and then we can see nerve invasion, some of the animals as well. So this model is very good because it mimics the disease, cutaneous SCC, with nerve invasion in the head and neck. Yeah, so I know that Shannon has already gone through our hypothesis with you all, but I'm going to go back to that and to conclude my talk. Summarizing, so we know that tumors, they can be resistant to monoclonal antibody therapies, and they can also acquire this resistance. So we have three scenarios here, and we have tumors that actually hold these targets inside. So we have internalization of drug target. So if the targets are internalized, we don't have these targets on the surface for antibody binding, so there's no immune response. So scenario B, we have drug target on the surface, however, they might not be enough, or they're not clustered together. So even though they have some interaction with NK cells, this is not enough to elicit a strong, potent immune response. In scenario B, and this is how our technology works, we add prochlorperazine or PCZ. So we inhibit dynamic in cells. Therefore, we can inhibit endocytic pathways that are dependent on dynamic, for example, clathrin-mediated endocytosis and fast endothelium-mediated endocytosis. And what happens is that we not only trap these receptors on the cell surface, but we can cluster these receptors on the cell surface. And when that happens, in the context of monoclonal antibody therapy, these antibodies bind to the cluster receptors, and we have a stronger interaction with the NK cells, as Shannon showed, and the EM. And we have release of the lytic granules and enhanced tumor cell killing. So I'm going to finalize this talk with this animation that I made to explain our technology to a general audience, but I thought it was kind of cool and I wanted to show you too. So basically, this is how our technology works. You have, oops, didn't go. Okay, so we have the complex antibody receptor being internalized. When we add PCZ, we see these receptors clustered on the cell surface for antibody binding, and we enhance interaction with natural killer, so these tumors are killed. Thank you very much. This is the amazing team we work with. So thank you, everyone. Thank you.
Brilliant. Thanks so much to you both. That was really excellent to hear about your work. Fiona, are you happy to go through the questions?
Yes. So the second one I just answered by chat. Yes, we do move the MHC class 1 and 2 receptors, but we know that that's not a short-term effect on ADCC, because if we use the K44A dominant negative dynamic mutation, we see the movement in MHC and tumor antigens, but we don't see a significant increase in ADCC. And so we're looking at those movements of receptors, but we think within the context of in vivo work, that's more likely to lead to longer term effects in memory. And it's not a change in the short-term ADCC. There's multiple mechanisms happening here. The other thing that we know from response to cell reviewers is that if we use on-target dopamine receptor inhibition, then we activate NK cells, but even though the NK cells are more active, it doesn't actually increase ADCC. So within the patients, we know that we probably have increased ADCC, ADCP, we temporarily inhibit AKT, we temporarily activate NK cells, and and there's multiple different mechanisms happening, but none of our patients, we don't think, would complain at multiple positive anti-tumor mechanisms happening at the same time. Fernando's asking about the idea that NK cells do anything at all in primary tumors. It's a long discussion we've been having with the drug companies over 10 years. One of the interesting things we've noted is that when Merck first produced avelumab, they were late to the race with the checkpoint inhibitors, and their assumption was that because they had an active non-silent FC region, that they'd have more efficacy because they'd actually also induce ADCC. And it doesn't really induce ADCC, which is odd because the IgG1 of avelumab is identical to cetuximab because they used exactly the same vectors to produce the antibodies. But if you use PCZ, you increase it. And so EGFR and HER2 all innately cluster because they hetero and homodimerize, whereas PD-L1 doesn't. And so we think the fact that they didn't see the ADCC was actually due to non-clustering of PD-L1. And when you cluster that, you improve it. So they've done a bunch of experiments, and we need to look at that because when they do their experiments in things like NK depleted mice, they're already not inducing ADCC. And so those experiments, as Fernando suggests, really need to be done in the presence of PCZ or something to cluster the receptors so that we can see the reality. But we also know that in all our models where the NK cells aren't active, we don't actually get the full anti-tumor effect. So we know there's NK-mediated effects in there. So it may be a normal monoclonal antibody therapy that the NK cells aren't being a significant part of it for various reasons. But if you cluster them and activate them at the same time and activate the NK cells, then it may be that they can be part of the immune response. And what we've noted throughout our trials is that these give you a particular effect, not so much in the primary tumor, but metastasis, which is really what we want to hit in our patients. And so it may be that currently NK cells aren't as active as they could be, and that we've just happened on a way to increase their ability to hit, especially metastasis. Is there any yes, so we looked at that, Dave Evans. So there's a lot of people who take lifelong antipsychotic PCZ. And so what it turns out is over a lifetime, they have a decreased tumor risk, but they have an increased risk of metabolic syndrome, which is probably due to the fact that every time their receptors are inhibited on a daily basis over many, many years, the insulin receptor will be shifted as well. And so they do have a decreased tumor risk. And one of the things before we started the trial was we got Merck to check their extreme trial, the original big trials for cetuximab, because people don't come off their antipsychotics to get cancer therapy. And so they were able to tell us that there were no unexpected side effects in people being treated with cetuximab who were also on their antipsychotics, but they didn't have enough numbers to tell us whether there was a positive outcome or not related to the psychotic. So yeah, there is epidemiological evidence over time, but PCZ on its own doesn't do it, and we see that in all our models. Can you do the opposite to the receptor expression at the cell surface? Increase internalization. So that's Ranjeni. So things like trastuzumab emtansine are designed to deliver a payload of toxin to the endosomes. And there's a couple of new papers in cancer discovery showing that increased endocytosis actually improves the targeting of these toxins. And so they've kind of said that that's, while we've got blocking internalization increases ADCC, whereas they show that increasing endocytosis improves toxin delivery. And so they've written them as if they're two separate things. But from the results that Priscilla showed you, this is a reversible mechanism. And so what you can actually do is build up your drug targets on the surface using endocytosis inhibition. But then as it releases and reverses, you get a wave of endocytosis. And so in that way, if you're using something like trastuzumab emtansine, you'd get decreased tumor heterogeneity, better drug binding to the surface, and then a massive wave of endocytosis as it releases, which would then deliver the toxins to the endosomes. And at the same time, you'd have enhanced the ADCC to the IgG1. So one of the things we're setting up to do is to try this mechanism with trastuzumab emtansine or some of the newer ADCs, because it's not, people always think in absolutes, but blocking endocytosis also increases at the end, endocytosis, when it releases, and exo and endocytosis are in balance. And so when you block endocytosis over time, you also block exo. And so you probably decrease things like IL-6 secretion temporarily. And then on the release, you reset. So one of the things we've noticed is that the targets of the drugs increase after the release. Now, it's been shown over many years by people like Dick Anderson that there are actually oncogenes expressed in tumors which hold receptors in the early endosomes and stop them from recycling to the surface. And so we think that when you block endo and exocytosis and then release it, that you actually reset the system. And some of those endosomally trapped receptors can then be recycled back to the surface, which makes them increased drug targets, basically. There's another question, Fernando. When you don't see ADCC but NK cells are active, could be because activated NK cells, Mr. CT. Yes, Fernando. So there's a lot of immunology to be done, and there's a lot of different mechanisms to be looked at on the actual endocytosis side, because even though we understand so many endocytic pathways in cell models, nobody's ever looked in vivo. In fact, the assay I invented to look at the surface levels in vivo of the tumors is the first one that's able to look at that actually in vivo mechanisms. And so we're just working that with people like Rob Parton and Jenny Stowe, and with the immunologists to look at the immunology side. There's multiple questions, but in the meantime, we're also setting up all the phase 2 clinical trials in different cancers with different drugs. And there's other antibodies to look at. So there's a new anti-HIV-1 antibody that's been developed where application of that monoclonal antibody to HIV patients means that they can come off of the proteasome inhibitors that they're on for years that cause a lot of side effects, and they stay off them for 5 years. And that turns out that anti-HIV-1 effect is mediated by ADCC. And so there's also other types of antibody therapies we can look at. But on a really basic level, and I think what people miss when they're talking about the NK cells and the ADCC is that that's in the context of an IgG1 monoclonal antibody therapy. What we really want to look at is the fact that when you use PCZ, you can temporarily move drug targets to the surface, and you can decrease heterogeneity in tumor cells. And so regardless of any of the immunology mechanisms, it may just be a general way of improving drug targeting into tumor cells temporarily. Does Ranjani immunohistology show you where the NK cells are localized in the tumors? Might need to. Yeah, we don't know. And I've just submitted an ideas grant whereby we want to take the murine and human treated tumors and actually do this new kind of electron microscopy whereby you do 3D serial sectioning through the tumor, and every second section is taken off to be labeled by light microscopy and then correlated back onto the electron microscopy. And so what we're planning to do is build up a 3D image of actual in vivo treated tumors from both the immunology and the cell biology point of view, using 3D electron correlative light microscopy, which is really cool stuff that Rob Parton does.
Might just take that one last question. I'm just time before moving into the morning tea.
Don't know, James, but we certainly know that if we don't have the IL-15, IL-15R receptor complex in with our PBMCs, that the NKs aren't working very well. But in humans, there's probably a lot of that there. I don't know. It's a good idea.